Thank you. Good afternoon, everybody, and welcome. And welcome to the last of our series, which we've called the Young Scientists series. This is an initiative of one of my colleagues, and just to interrupt myself and go back a step, what we do, part of our summer school work, is to showcase the work of the university, to give the public, which is you, access to the most recent research. And we try and get where we can, like in the science lecture, younger lecturers, and give them exposure to lecturing. But most importantly, it's to showcase the research of the university. Now, the young people like our lecturer today and the two that we've had previously are young scientists studying at this university who may or may not become academics. But I think it's a wonderful initiative for which I thank my colleague, and we've had fantastic feedback about the lectures, and I hope that this one, I know this one won't disappoint as well. It's such a fascinating topic, and you've also got a young lecturer who has an impressive CV. So let me tell you about Jessica Dawson. She has a BSc in Marine Biology and Oceanography, a BSc Honours in Zoology, and an MSc in Marine Biology, all from this university. She's currently working on a PhD in estuarine ecology at UCT. She has won the class medal in her honours year, has been on the Dean's Merit List, and is a member of the Golden Key International Honour Society. Now, to be on the Dean's Merit List and to belong to the Golden Key, you have to do exceptionally well. Jessica has written a number of academic papers on topics such as starfish grazing and crown crabs. It's a long way from hippos. She has presented posters and papers at workshops and conferences and has attended the Côte Summer School in Bordeaux in France. Jessica has also presented several guest lectures in the Department of Biological Sciences. Please welcome our young scientist.
Thank you very much. Apparently I'm a little bit low position, so if you can't hear me, just let me know. Thank you. So I'm here to tell you how I got to where I am, what I did, and what made me decide to come to where I am, and a little bit more about my PhD and what I will be doing. So as they say in Alice in Wonderland, we begin at the beginning. I'm one of 4 kids, and my mum did not want girls. She said they were full of fimmies, full of nonsense, much too prissy, and generally too scared of things. I think she regrets this a little at the moment, but after the second boy, she was very welcome to have girls, but decided to make sure that I never had any of those characteristics. So when I was very small, one of her favorite topics is that she got a phone call from the neighbor to say that he'd heard me cry and stuck his head over the wall, and my 2 older brothers were teasing me with a frog. My mum told him not to worry. She'd been out to see what the commotion was about, and I wasn't actually crying because they were teasing me. I was crying because they wouldn't give it to me. So this is how things came about, and how my love for nature developed, is all these things that happened in my childhood. So learning how to fish at an early age, still fishing, apparently having a very great affinity for mud. This one has also not changed. You know, who needs a spa day when you can just go out and collect samples? The one my mum regrets most, I think, is that upon finding a chameleon, my cousin and I decided to do a complete project with images, color changes, and full description of life history on this specific reptile. The next reptile's a little bit bigger, a little more scary. So growing up, my mom was not allowed to have animals in the family, and I think she made sure that this changed. My dad always had cats and dogs, but we had cats and dogs. We had ducks, chickens, geese, baby rabbits. The mother died when they were 2 days old, and I ended up feeding rabbits with bicycle tube and a syringe for every 4 hours for about 3 weeks until they were big enough to eat on their own. She rescued chickens from the side of the road and decided, here, you're studying zoology, raise these. In the end, they followed me around the garden for several days. But this was the general state in which I grew up. So my big decision was I was going to be a game ranger because of all Kruger Park visits, because of my grandfather's love for giving us National Geographic's videos, or I was going to be a veterinarian. And then all of this changed when I was about 15, and a friend introduced me to the idea of scuba diving. So my dad, being a very avid snorkeler and managing to stay in the water longer than his kids, even with blue lips, decided this was a great idea. My mum always keen on encouraging us to do whatever you want to do, and that is to your ability, said go for it. So in 2002, I got my scuba diving license, and since then have got another 3, including my rescue diver and my commercial scuba diver's license. And unlike Alice, I don't have to go down the rabbit hole. All I have to do is go underwater, and it's a completely different universe. And this is where my love for all things marine developed. I wanted to look into it. I wanted to know more. I wanted to find out why, where, because of what reason. And this is what started my undergrad career. So I came to UCT to do marine biology, and combined that with oceanography, and as a added extra, because marine biologists talk to dolphins, I took an extra year of maths, physics, and chemistry, so I can teach as a backup. And then in the second year, I met the most amazing prof. He's not got anything but grey on his head and still has the same enthusiasm that I had at the beginning of varsity for a subject that he's been doing for 20 years, more than 30, 40 years. It's fantastic. He has this phenomenal enthusiasm, and he encouraged me to go further and study further. Because of this, I ended up getting the nickname Decade Dawson, because I was going to be that long in the university. Currently, I'm on my 11th year, so they weren't wrong. And it was thanks to his enthusiasm and his encouragement, and seeing how much listening to him made me want to know and learn more, that made me decide I wanted to go into academics. I wanted to become a lecturer, become a supervisor, and if I could instill even a portion of the enthusiasm that he instilled in me in any other student, I'd be very grateful. So to do this, I did an honors project on crabs, in which another student had discovered that there were multiple different species, and I had to describe what the difference between these species were. So you do fine-scale line drawings and a good description of the slight differences and how you would identify them separately. I then moved on to do a project on starfish and expanded on this in my master's year. So we're putting cages in the soft sediment environment in Langebaan, and then we monitored over an extended period of time how different numbers of starfish would affect the microalgae and the macrofauna within the system, and what effects they have, and what would happen if the numbers started to drop too drastically within the system. So we just needed to get a better understanding of what the starfish were doing within the system. From there, big leap, I know. People always ask me, how did you go from starfish to hippopotamus? Well, grazing in starfish, grazing in hippopotamus, definitely the same thing. Turns out my supervisor had a dream about hippos and crocodiles, and decided that very little research has been done on these animals in South Africa. And he came up to me and he said to me, look, would you like to do this project? It's great for a PhD, it's very unique. And I jumped at the opportunity. I'd never been to St. Lucia before. I wanted to see the area and figured that it would be a very exciting, if somewhat tricky, thesis. So what do we know about hippos? One of the key things is that they are only found in Southern Africa. So they're not found on any other continent, which makes them quite unique and gives us a nice sort of niche in which to study them. They wallow fantastically during the day, very lazy animals during the day, but then move out onto land at night, and this is where they consume the food. So hippos being very large animals, the males can get to anywhere from 2 to 3 tons, which you can't quite fathom until you've seen one. You know, one of them walked past my window one night, waking you up in the middle of the night, and it's at least like the size of both tables, and you know, flush with my head in the pillow, and it's slightly alarming. But enormous, enormous animals, so it needs a lot of food. So they can consume up to 40 kilograms of grass in one day. So then, what makes them an interesting topic, aside from the fact that so little has been done on hippopotamus? And most of what has been done is limited to captive hippos, hippos in zoos, or hippos in very small, shrinking dams or man-made dams. So this posed a very unique opportunity because we could study them in the wild. And because of this lack of research, very little is known about hippos. But they're one of the largest animals found in the estuary in St. Lucia. So it also creates an opportunity for us to assist in conservation matters, because the more you know about the animal, the better you can conserve or monitor what it's doing to its environment. So of the estuaries in South Africa, there are about 250 estuaries. St. Lucia is the only one with a large, sustainable population of hippopotamus. They've tried to introduce a small group into an estuary close to Grahamstown, and the 3 that are supposedly in Rondebosch really don't count. In Rondevlei, it's not quite enough for my liking. So one of the really interesting things about the hippos, and the actual crux of my thesis, is the fact that they feed on land but live in the water. So they create this bridge. They're the only mega herbivore, or big herbivore, that moves between the terrestrial and the estuarine area. And this means that they're moving a very large quantity of terrestrial material into the system. So my supervisor likes to tease me that I have a very crap project, because this is my thesis. I want to know what the effect of the hippo dung is as a transfer of energy from a terrestrial environment into the estuarine system. So I said that hippos consume about 40 kilograms a night. Of this, only about 10% is ingested, and the rest comes out as waste. Of that, about 20% goes onto land, and the remaining 80% is then excreted in the water. So if you consider that, sort of taking a low estimate of hippos numbers in St. Lucia, there are about 900 hippos in the system. This amounts to a total of 28,000 kilograms of dung in a single day. That's 28 small cars. I mean, it must have some impact. It must be doing something, and we want to know what. So my study site is St. Lucia in KwaZulu-Natal. It's the largest estuarine lake in Africa, and consists of 3 shallow lake systems in the north, and then a 20-kilometer section here that is called the Narrows, which is the main area where you find the hippos in this system. One of the key things about the system is because of droughts and a lot of things, a lot of farming and water retention elsewhere, it's one of the largest protected water bodies in the area. So more and more hippos are moving towards this area and using it as an important resource. And therefore we need to know what the impact of this is so that we can monitor and control and protect the system. So my research is done in the south. I have 2 main sites. The one at the bottom here, which is where the hippos generally occur, and then a site higher up, which is for comparative purposes, where there are very few hippos and no permanent pods. And there are 2 sections to my thesis. The first one is trying to determine and find the dung in the system, trying to see how far it travels and how high up the food web it's going. Is it an important resource? And if yes, what is it doing? So in order to do this, I go out and collect samples for the entire food web. I collect muddy cores, for the microalgal samples, so that's the primary producers within the system. It also gives me a measure of what's in the sediment in terms of organic matter. I also collect water samples to have a look at what's floating around in the water column. We use this fancy thing called a D-net that we sling behind the back of the boat, and that collects the zooplankton within the water column. And this is one of the middle stages of the food web and is quite an important food source. And that is then filtered and can then be analyzed. The muddiest one is we use this, which is called an Ekman grab, to collect samples of the sediment on the bottom. We sift this, sort through it, and come out with the different forms of macrofauna in the system. And the final one is to collect the dominant fish species that are found in the different areas within the system. And this helps us build a food web of everything from bottom to top. Now, just to make sure my friends aren't too jealous, I have to do some tedious lab work, analyze all this data for isotope and fatty acid analysis, and then use this to produce the food web. So this is a very basic idea of what I'm going to get. This is a food web if we do not have dung. So this is taken from the north site. It's an average of my results. And this is what it would look like if you don't have any dung in the system. If we add dung to the system, you instantly see this shift from the benthic organism, so what's on the bottom of the water, and what is generally the primary food source, has this distinct shift in its signature. And if you then look at the food web with dung present, the whole food web has shifted. The whole food web changes. So what this is telling us is that the dung is having a distinct effect and is found throughout the food web. So some species, like mullet, which is a form of fish, are known to sit behind the hippopotamus, and his last meal becomes their next. In other cases, there are carnivorous fish that are not eating any of the material, but because they are eating a smaller fish that has eaten the zooplankton, that has consumed something from the bottom levels, the dung signature can be seen throughout the system. So this shows us very nicely that the dung is important and the dung is having an effect. So the next step was to get very fancily dressed, to go out and to find out what effect the dung is having on the benthic community, so the animals found in the sediment, in and on the sediment. In order to do this, I went out and set up a number of cages, so that, because it's very new research, we did a very basic experiment where we were controlling the conditions within the system, and then adding dung to half the cages and no dung to the other half. And at the end of this, we could then collect samples to see what the difference in the community is based on having dung or no dung. So in order to do this, I spent a number of early mornings walking along hippo paths, or hippo highways, as I like to call the one that is that distinct, looking for very fresh hippo dung. And in fairly large quantities. I had to have the game ranger with me because they said that you needed someone with you, but he only carried a camera. So decided I just had to be able to outrun him. That being said, a hippopotamus can run 40 kilometers an hour on land. I can't really do that on my bicycle unless I'm going downhill. Thankfully, I never came across one. Then I would go every week and put a measured amount of dung in demarcated cages and make sure that the cages were still properly set up and controlled, and that I always added the right amount of the same amount of dung to the same cages. The man who invented cable ties is magical. The woman who invented multicolored cable ties, definitely a fan. So going back to what Mom taught me in life, fertilizer should have a positive effect. It did in her garden. Add a little bit of science to that and a lot of talk between my supervisor and I. And we assumed that the addition of hippo dung would have a positive effect. It didn't. So all of the characteristics that we went and tested, after collecting samples after 6 weeks, I had benthic samples for chlorophyll A, which gives us a measure of the primary production. I had myofauna samples, which is the small animals that you found between sand grains, and macrofauna samples. All of them showed a decrease. Not always as strongly, but they did show a decrease. And this is very interesting. It's not what we expected, and it brings about endless number of more questions. And this, to me, and my supervisor, is the excitement of research. The more I know, the more I learn, the less I know, my mum always used to say. So it's just asking a lot more questions. So moving on from a bit of the science, not that I did too much, I hope. These are just a couple of anecdotal stories that happened during my thesis, a little bit of things that I came across. When I first started, I made sure to go and do my research and ask everyone in the Parks Board, what do I need to worry about? How can I protect myself? Given that I am standing in fairly muddy water, knee-deep, surrounded by crocodiles. The croc expert says to me, don't worry, don't go deeper than your knees, keep a lookout, carry a stick. If it comes close, bang it on the head. I had a very sturdy stick. It went with me every time. Thankfully, didn't have to find out if banging it on the head works. So, Murphy's Law says when you're looking for one thing, something else will appear when your parents are visiting. So completely sort of unexciting, not nerve-wracking experiences until mom and dad come to visit. And I'm standing putting in my samples, and the one set of cages I had, as you can see here, are next to a management jetty. And so I was looking up, making sure that I had seen no crocodiles, and this very distinctive sign of other danger appeared. I made a slow and very ungraceful attempt to get out of the water on sort of chest-high jetty. My mother was laughing, taking photographs until saying, "What are you doing? You're not finished." "Yes, mother dear, but right now I'm trying to get away from a shark." She stopped laughing quite quickly. It was fantastic. It was great. I hadn't seen the sharks. I knew they were there. I was hoping to be able to get some samples for the sharks, but we didn't manage to catch any. It was a small shark. It was about 1.5 meters, and it swam around for about 20 minutes. After which time, my mum thought it was very amusing, after trying to convince me I didn't need to go back in the water, and me telling her that it couldn't come close without me seeing it. I needed to finish putting my dung in my cages. I needed to finish doing my work. She thought it was very amusing that I was about to climb back into water with a shark currently visible, but first had to put on my gloves. So the reason for this that I then told her is that in the entire time I've been doing my 2.5 years of research, the most dangerous thing that has chased me is a dung beetle. So hippo dung is, I might sound crazy now, but it really doesn't smell that bad. It's quite a fresh smell, it's a grassy smell. However, the smell does linger. So after a sampling day, I went to the beach to watch the sunset, and the entire walk from the car to the spot I stopped, I was making sure that I didn't get a dung beetle alighting on my head. The other exciting part of my thesis that was very different and didn't have to be analysed too hectically was catching crocodiles. I only told my mum about this after the fact, sent her some lovely photos with a disclaimer in the title line that please remember is afterwards and I'm currently safely in Cape Town. So how do you catch a crocodile? We go out on a small boat at night and you shine a torch in the water, and they have very reflective, very red eyes. And this enables you to see where they are. So you slowly motor up, cut the engine, and drift towards the crocodiles. Thankfully, the researcher hadn't been doing croc capture for quite a while, so a number of the crocs were far more relaxed about the situation. That being said, the first time I tried with the noose, it was me who wasn't relaxed. I saw it, decided it was too big, I didn't want to fight it, and I faltered and it disappeared. But what you have is this long swimming pool pole with a metal noose on the end tied on with masking tape. And you lower it over the crocodile's head and you yank the rope and release the pole at the same time. What follows is about 5 to 10 minutes of spiraling and splashing and quite dramatic activity. And then you pull the crocodile up next to the boat. Once you do this, you have a nice pole with a cable tie attached with a string through so you can pull it close at a safe distance, and you close the mouth. We then move slowly to the shoreline, and you pull the crocodile up onto the shore. And this was my first capture, and many people say I look both shocked and very excited in my photos, but it was amazing. You never quite have the respect that an animal deserves until you've seen it up close. And it's a phenomenal creature. Terrifying, but phenomenal. And I got a full history. We did measurements, we sexed it. We then got a great description, as the croc expert gave me all the details and did a very nice, basically mini lecture for me so that I could learn a bit more. And then I collected my samples. How do you hold down a crocodile? How do you keep a crocodile? You sit on it. The bigger ones, at least. Something about having the weight on them apparently prevents them from getting too excited or thrashing or moving. So once we pulled them on shore, you cable tie the mouth, you double cable tie the mouth if it's a very big one, you duct tape the mouth, you cover the eyes because it protects them from the light, as well as calming the crocodile. They stop moving almost instantly. And then you also tie up the back legs. So the back legs are their powerhouse, and when they move away, you see them kick off right from the back. So if the legs are tied up, it prevents them from moving. This was pretty important in this case because we couldn't get the crocodile out of the water. It was too heavy. There were 3 of us, and we estimated that it was a little over 200 kilograms. And it just sat, like a car stuck on the beach. It wouldn't go anywhere. So we were standing in water about 6 inches deep in an area where we just counted 17 crocodiles. So my 2 field assistants who were with me took a look at me and said, "It's your project, you're getting in there, not me." And they were boys. But anyways, another way we caught a crocodile was a crocodile that was being a nuisance, kept going for this, the boat captain's broom. Whenever he'd washed his boat and put the broom in the water, it was biting pontoons, it was disturbing the passengers as they were moving in and out to get to the boat. So we put the broom in the water with the noose around it. As the crocodile came up, we grabbed it. This posed a bit of an interesting question as to how to transport it, because it was an unexpected call, and we made a plan. So, this is Africa, let's put a crocodile in an Ivanza. The second one we moved was because of the drought. About a week before, they'd caught a crocodile walking into town. So it was looking for fresh water, and obviously it's not one of the things you want in your garden pond. So they called the park sport, and the park sport collected it, and then because they didn't have it and it was unexpected, they put it in one of the holding pens within the croc center. So we went and gave me a nice sample of a crocodile, and we caught it and then had to move it. So we now had 2 crocodiles, thankfully on the back of a bakkie. And again, it's like tricky transporting matter. So we moved them to a lake further north because crocodiles, if put higher up in a river stream, will find their way down eventually to where they were. And therefore, we had to put them in an enclosed system. But the closest road was 700 or 800 meters from the water's edge. So thankfully, I only had the tail, but each one of these crocodiles was from about 120 to 90 kilograms. So it's a slightly awkward, unexpected weight. So this is also an interesting story, and one of the crocs we caught. This is a small crocodile, and still could not be held with one hand. So it was still strong enough that wriggling in my hand with both hands, I had to hold tight. But when I collect samples, what I do is cut off a tail scoot. So down the back here, they've got tails on left and right, and then down the middle. And we remove the scoots. That way I get a tissue sample, but you also mark the individual. This individual was already marked. So it had scoots missing. So the croc expert who's always with me went and looked up his research and discovered that this crocodile, a young female, was born in the croc center, was tagged at 1 year old and released into the system. And we caught her almost 6 years to the day after she'd been released. So in terms of science, this is a fantastic one because you can then go back and see, are the conditions good? Is she growing as fast as she should have been? One of the big things that people seem to forget when you say you've been catching crocodiles is, how do you release it? So I just poked around, aggravating a crocodile. How do I let it go? This was the most difficult one. This is a 3.4-meter crocodile, a very large male, close to one of the largest ones they found in this estuary, although they can get to 5 or 6 meters higher up in Africa. It's very big for South Africa to be 3.4. So it was really well trussed up and holed, and now you need to let it go. I've just cut off 3 pieces, 3 scales, and hoping that it's not too upset with me. And it took 3 people. So first you cut the cable ties. Then you loosen the back legs, and in this case, normally you have one person hold them up and out the water, so they still don't realize that they can put down and go. I couldn't hold 2 legs. Gave it over to the rugby-playing gentleman who was with me. He couldn't hold 2 legs. So we each grabbed 1, double-handed back legs. Then the croc expert, who's much more comfortable with the front side of a crocodile than any of the rest of us, goes and he puts a piece of rope that has a carabiner around the duct tape, cuts the duct tape a little with his Leatherman so that it's starting to tear, and then on 3, grab the towel covering the eyes, back away, and at a safe distance, yank. The tape tears and the crocodile is free. This one sat still. So he was going nowhere. Boat was right there. We waited for about 10 minutes before it slowly moved off and we could get back in the boat. So, fantastic experience. Amazing. All these things that I've done as a kid when I'm very small have impacted what I've become as an adult, which made me think, what do you think these kids are going to do? So these are the 3 sons of the croc expert, and my first croc, the dad says to his boy, why don't you just sit on it, just keep it there for a while, we're going to go. And 10 minutes later, he's completely bored. And this is the 6-year-old son who came with us the first time, and also quite comfortable, standing holding dad's rifle while drinking Oros. So then after this, I've done all my sampling now. I'm busy writing up. I will hopefully be able to complete my thesis by June. So what's next? Ultimately, I want to be a researcher. I want to be a professor in the long run. And I don't know what my next topic will be on, but I know that I want to do a postdoc, which hopefully something overseas, maybe something that links into animal paths. Hippos have amazing paths that include underwater, and we got this very fancy device to be able to see if we can follow them. But my thesis got a little bit large, and so we couldn't do that. So the only thing I do know is that I'm going to continue with what my parents taught me about making sure that wherever you go, you try and learn as much as you can. So an example of this was Christmas, slightly different animal capture, but I heard that I could go and help and assist with a vet who was helping them move 3 nyala that had escaped back to the farm where they'd come from. And so we had to walk around in, I think, 33 degrees. And once he darted the buck, we had to follow it and make sure we could catch it as fast as possible and move it up, because nyala are prone to having heart palpitations if they darted and lying down. Put them in a sling and then transport them to the local farm where they belonged. Another thing that hasn't changed is a love for birds, more so since going to St. Lucia, because of the amazing species that are there. And because I've always lived in the high veld, and then here in the Western Cape that has a bit fewer bird species and a lot more LBJs, which are a lot harder to identify, suddenly there were these birds that looked tropical, and I just exploded in my interest for bird life and trying to identify them, and have recently joined a group that go on a monthly basis to ring birds. So again, just constantly asking questions, trying to find answers, and trying to see where that'll take me. Thank you very much. I'm happy to answer any questions if you have any. Yes?
In the estuary in St. Lucia, when you were doing the hippo dung cages,
Yes.
that you put dung in some and some not, and no dung in others, was there a tidal effect there? Did that skew your project a bit? Or
No.
were you far away from the tidal
So,
impact?
at the moment, St. Lucia has very little tidal effect whatsoever. It tends to go through cyclical periods of drought and flood of anywhere from 8 to 10 years. It is now in its 14th year of drought. So they had some rain, but since 2002, the mouth has been closed with only some release, when 3 years ago they opened a small back channel from the adjoining Umfolozi River. So in general, it has very little tidal action. We also chose a site that was away from hippos, that was sort of limited differences between the narrows, but that also where we could, as best you could. I mean, it's a problem with all in situ experiments, any experiment in the wild, because you can't control everything. But that's why we had repetitions, and we kept it at a level where hopefully the conditions are kept quite stable. We have theorized that at one set there was a bit more wave action than the other, and that might have account for some of the differences we see, but all of that is only observational.
So the sharks that were there, they weren't coming in from the sea. They have at some point come in from the sea, and they simply breed and live there.
Yes. So it's like the Breda River. The Breda River has a number of shark species. This is the bull shark, or the Zambezi shark, which is known for wanting to breed in nursery environments. So several years ago, the sharks would have been coming in, and then they breed within the estuary, and the adults might move out, but the youngsters then stay there. So now, because the mouth has been closed, they have quite a large population of sharks, some of which are growing quite big, that you can see.
Eating fish or game?
Fish. So very rarely you would find sharks eating game. There's some very cool photos of a crocodile eating a shark, but generally they're just fish. Yes?
Do hippos have any predators?
Not really. So there are a couple cases where they find lion or something, or crocodiles predating on juvenile hippos. But most of the time they say that the chances are the hippo had died already. So, I mean, a young hippo weighs more than me when it's born. So they weigh anywhere from 60 to 100 kilograms, the newborn hippos. But also, I mean, the mothers are very territorial and very protective, and the hippos stay very close to them. So one of the reasons why hippos are one of the most dangerous animals is if you become between a hippo and her baby, she makes a beeline. And they don't eat people like a crocodile might do, but they will trample you, and at 2 tons, it's a pretty dangerous experience.
Why is the hippo not part of the big five?
Oh dear, I have no idea. Perhaps because people hadn't originally seen them so easily. I mean, all the big five are quite picturesque and quite easy to see. Hippos, you don't often see them out of the water, so I mean, just seeing the nostrils, they don't look quite as exciting, as intimidating as a rhinoceros until you see them up close and outside the water. So, yeah.
What's the prospect of getting hippos in all those other estuaries you mentioned, in particular where I live near to one called Sunflag?
Yes. Apparently the hippos have sometimes moved there. The rondaflay hippos have sometimes been seen there. I haven't even seen the rondaflay hippos. It's very unlikely. So in St. Lucia, it's a very small town. the hippos still walk out and go and graze on the lawns. I mean, there's signs everywhere that say, you know, holiday people, please be careful. The hippos come out. We're in their environment. Respect them. You drive very slowly on the roads. They wander around the town because it's nice green lawns. So that's one of the biggest problems about trying to relocate hippos into other estuaries, is that there's human population around most estuaries. And I mean, my mother was terrified. What are you marine biology, what are you doing hippo research on? They are classed as one of the top 5 most dangerous. I think they're actually number 2 after the mosquito. So the most kills in Africa is the hippopotamus, in wild animals. So it would be very difficult to convince people that to let hippos into other estuaries. Sorry.
You indicated that the population was declining. Is there any chance of them becoming threatened in due course?
No. So at the moment they are approaching threatened outside of South Africa and in some areas in Africa, simply because of human conflict, so humans moving into the area. In South Africa, with the Kruger Park and with St. Lucia being such a protected area, our populations are actually increasing slightly. So our hippos in South Africa are doing pretty well, but the rest of the Africa not so well. Yes.
What is
sorry.
the dentition? I mean, how do they eat the grass? I mean, you were talking about grazing it. They couldn't mow lawns, but I suppose...
They do. They do. We call them hippo lawns, like you call them rhino lawns, where you go walking, you follow this path and you stop and you suddenly, beautiful grass. And because of the certain type of grass that is obviously more enriching to them, they crop it, and it's a grass that then sort of thrives on this. So some of the other grasses that would be in between there don't come back, and they make these beautiful lawns, and that's why they have these distinct pathways or highways heading back to these lawns. So they have big teeth and much more like molars, so much more like your back molars. They're very big canines are for show and for the males for fighting, but alternately they have very flat molars, and that's another research opportunity, is to look into their teeth, because that the enamel and all of that saves a history of what they've been eating.
What effect did you find that the dung had on the estuaries?
Well, in terms of that complete shift of the food web was a very interesting initial effect, and the opposite of what we expected in an increase of fertilizing effect is that we had a negative effect. So unfortunately, because they're so little studied and so little has been done on the hippos, that's the only thing. I mean, that took us several months, but it's a start. It's a baseline that says when you put hippo dung in the quantities that we found in the narrows, where there are large populations of dung, if you repeat that quantity in a small area where you don't have hippos, it has a negative effect on the organisms there. So the next question would be, if you controlled the amount of dung, what would the effect be? Does it actually completely change the species, or just decrease them a bit and create a more broader network of species? So it means more experiments, more time, more questions, more desire for research.
You never said what you found from the crocodile.
Oh, unfortunately, yeah, I haven't had that data analyzed. It's in the process, and the reason we also only had so few crocodile tissue samples is that you can't get crocodile samples at every single site. So because they're such mobile animals, we just wanted an example. And I got 7 crocodiles, but unfortunately I haven't yet completed that data. So
How does starfish do?
how do they do?
How are they doing?
Oh, the numbers were declining quite significantly since a big boom that we had about 10 years ago. They slowly decreasing, and part of that is that the seagrasses in Longabon Lagoon, which is a worldwide phenomenon, is the seagrasses are depleting. And although the starfish don't often occur within the seagrasses, a lot of the detrital matter that comes off the seagrass acts as a food source. But these starfish have a very interesting effect in that the way they feed is by extruding their stomach onto the the sediment, and therefore leaving like a slug would. They leave a mucus trail wherever they go. And that makes like a sticky glue that allows any kind of other organism that is, or small things that need to have a larval phase, to stick and then start to develop. So it's kind of like fly tape for food for other organisms. So the starfish have a very positive effect until the numbers are too high, and then are slowly having a negative effect when their numbers are too low. So it's a very classic shape in science, or in environmental science, that at the optimal density of starfish, you have optimal conditions for everything else. So at the moment, I mean, this was about 2-3 years ago that we did the study. The starfish are fairly stable, but with the decrease in the seagrasses, the assumption is that they might decrease further, which would then alter which consumers are found in the system. Do you know, I'm not quite sure. I would imagine that they might be, just because hippos can't swim, so many people don't realize that they walk on the bottom, and then they have, like a fish has a bladder, an air bladder. They have the ability to raise themselves to breathe. So that's why when you see a pod of hippos, you often find that the baby has its head on the mother's back. And that's because that's the only way it's staying afloat. So I imagine they could be born on land and then slowly move into the water. They are born in the water, too. Oh. Nice. Thank you. I didn't know that. That's fantastic. I do know that hippo's milk is pink. Strange facts you learn. It's apparently very pink and very, very rich. Yes?
Hippos are quite famous for going walkabout. So I'm wondering, here at St. Lucia, you tell us there's been no rain for 14 years. Are the rivers still flowing, and what would be the factors that would stop them going walkabout?
So it's been in a drought cycle. They have had some quite nice rain, and the system has come up and gone down. So when I first visited in 2012, they'd had quite a lot of rain, and the water increased by about 0.5 meter in some spots. But on the whole, they've had far less rain than usual. But what it means is actually a lot of the hippos are coming towards the estuary, because although they need fresh water to drink, and the estuary is very saline, they can get the fresh water in the small ponds and things, but they can't wallow in those. They get stuck in the mud, or it's not deep enough for them to be able to completely covered, and they get very badly sunburnt. So that's why they need to be covered in water. So the fact that the estuary has that deep narrows channel means that they can go there and wallow there, and then they just have to walk further to find fresh water. So generally what would make them go walkabout is the search for fresh water. Yes?
When we went to Malawi, we were ordered not to walk along the dam because
Yes.
they ordered us not to walk. But when we got into the dam, we went to swim. We swam with the hippos.
That's brave. The closest encounter I had was a hippo that walked out close to my cages. Sorry, you're welcome to go. It's not a worry. I had come to put in dung, so I'd been in the morning, done my walk, and had my dung. And a large male hippo suddenly rose up and walked within 100 meters of the cages. Thankfully, it was as I arrived, so I saw him. But in order to get to my second set of cages, I have to walk about 150 meters along the water's edge. And the biggest danger is coming between a hippo and the water. So I went home, did the dung collection again the next day, and put the samples in the next day. Rather safe than sorry.
Just out of interest, hippos excrete all this dung into the water.
Yes.
And there's loads of hippos and loads of dung. What is the effect? You looked at the effect of the dung in terms of the fish and all that sort of thing. But what is the effect of the build-up of the dung?
Well, that's actually one of the big reasons why we wanted to know in that quantity what it does, because we took the quantity, the amount that we found in the rest of the system. And a big part about the St. Lucia is because it's closed, because there's very little fresh water flow into the system, and therefore the system is quite stagnant, you are getting a buildup of this dung. So unfortunately, we couldn't quantify that at the moment, but we did the baseline study to say, look, the dung in the current quantities has a negative effect in an area where there's no dung. So from there, we will postulate further questions and be able to do other experiments that say, what if it continues? Because some of it washes onto the shore. So if you have enough wind, if it's quite fresh, then it floats a bit before it sinks. And if there's enough wind motion, you get these huge swaths of hippo dung all the way along the edge of the water. But it must be having an effect.
Well, you could actually clog up the system.
Yes. But that being said, I put dung in once a week. By the time I came on day 7, that dung was gone. So, I mean, there are organisms that break it down, that are using it as a food source, and that break it into small enough pieces that it becomes washed away and can be available as a food source. Again, all of this is postulation, it's not... Yes. Yes. It will. My supervisor is very keen on trying to understand this. I will hopefully be able to. It's mean that you've got this great interest, and every question I answer gives me 7 or 8 more.
I have a personal experience that I don't only defecate in the water.
Yes. This is about...
About
20% is on land, because, I mean, while they're walking back and forth, they still defecate on land.
I read somewhere that only the males that defecate on the land and the females only the...
The males use it like rhino spray. So when the tail is doing this, it's spreading the hippo dung. So it's a form of territorial marking. The females, when I found samples on land that were most likely female, it looks like elephant dung. So it's balls of dung. So if the need arises, they will do it on land. But just because of digestive processes, most of it is in the water.